Hello students and welcome to this lecture on autonomic nervous system, one of the most fascinating part of human body. I am Harshit Kanchav, assistant professor at Lloyd Institute of Management and Technology, Greater Noida, and today we will driving into how our body manages to keep itself running smooth without you even think about it. From controlling your heartbeat and digestion to regulating your breathing and even your stress responses, the autonomic nervous system plays a critical role in maintaining balance or what we call as homeostasis. In this session, we will mainly talk about sympathetic or adrenergic nervous system and the drugs acting on it. So before starting, just have a look on what is nervous system. So nervous system is mainly made up of brain, spinal cord and nerves. So the major function of the nervous system is to control and coordinate the action by sending electrical signals throughout the body. If I'm talking about the classification and the types of nervous system, the major classes are of two types, that is central nervous system CNS and peripheral nervous system PNS. Central nervous system contains brain and spinal cord, whereas peripheral nervous system contains all nerves and sensory structures outside of the brain and spinal cord. The peripheral nervous system is further divided into two parts, that is somatic and autonomic nervous system. Somatic nervous system responsible for voluntary control of skeletal muscles, and autonomic is mainly responsible for involuntary control of glands and smooth muscles. ANS is further divided into two parts, that is sympathetic, responsible for fight and flight responses, and parasympathetic, responsible for digest and rest functions. If I'm talking about autonomic nervous system, it controls and regulates the internal organs without any conscious recognition or effort by the organism. So basically, it comprises two antagonistic sets of nerve, that is sympathetic and parasympathetic. Here we discuss the adrenergic nervous system. Adrenergic nervous system is a group of organs and nerves in which adrenaline and noradrenaline are released as neurotransmitters. These neurotransmitters are basically target the adrenergic receptor and produce a biological functions. The drugs which mimics the activity of this sympathetic nervous system is called sympathomimetic drug or adrenergic drugs. Basically, these drugs binds with the adrenergic receptors and produce their biological activity. There are a great number of drugs available which can affect adrenergic receptors. Example, dopamine, noradrenaline, adrenaline, isoprenaline, etc. The major function of the adrenergic nervous system is to produce fight and flight responses. For an example, if you sense danger, your heart rate increases, BP rise, eyes dilate, blood sugar rises, bronchiole expand, and blood flow shift from skin to skeletal muscles. Basically, this function, this adrenergic system is become active when you are in stress, trauma, fear, hypoglycemia, cold, or exercise condition. If I'm talking about the adrenergic neurotransmitters, these are the derivatives of catecholamines. Here you just see the structure of catechol, in which benzene ring contains two hydroxyl groups, which is attached with the ethylene bridge to a amine moiety or amino group. Okay, so you just see four types of neurotransmitter, that is dopamine, noradrenaline, adrenaline, isoprenaline. One by one we can discuss their structure. Here in dopamine, catechol moiety is bind with the amino group along with the ethylene bridge. Okay, in noradrenaline, the first carbon of ethylene bridge contains the hydroxyl group, as you can see in the structure. In adrenaline, the amino group hydrogen is replaced by a methyl group, and in isoprenaline, this hydrogen is replaced by a isopropyl moiety. So the structure of these catecholamines are somewhat similar, as they contain catechol moiety along with a amino group. So students, just have a look on biosynthesis of catecholamines. The biosynthesis of catecholamines begins with an amino acid called tyrosine, which can either be obtained directly from the diet or produced in the body from phenylalanine. This is the starting substrate for the entire catecholamine pathway. The first and the rate limiting steps in catecholamine synthesis is the conversion of tyrosine to L-DOPA. This reaction is catalyzed by the enzyme tyrosine hydrolase and it requires tetrahydrobiopterin as a cofactor. Because it's the rate limiting step, it's tightly regulated by feedback mechanism, especially from dopamine and norepinephrine levels. Next, L-DOPA is quickly converted to dopamine by the enzyme aromatic L-amino acid decarboxylase, also known as DOPA decarboxylase. This step occurs primarily in the cytoplasm of the neurons and is crucial in dopaminergic pathways like those in the substantia nigra and ventral tegmental areas. Now, in neurons that synthesize norepinephrine, dopamine is transported into vesicles where it is converted into norepinephrine by the enzyme dopamine beta hydrolase. This reaction takes place inside synaptic vesicles and requires ascorbic acid or vitamin C as a cofactor. In cells of adrenal medulla or central areas of the brain where epinephrine is needed, norepinephrine undergoes one more transformation. It is methylated to form epinephrine by the enzyme phenylethanolamine and methyltransferase or PNMT. This enzyme uses S-adenosyl methionine SAM as the methyl donor. Interestingly, the expression of PNMT is increased by cortisol, linking adrenal medullary function to the adrenal cortex. To summarize this up, the pathway is primarily start from tyrosine, which convert into L-DOPA, then to dopamine, then to norepinephrine, and finally epinephrine. Each step is enzyme catalyzed, requires specific cofactors, and occurs in specific part of cells, reflecting the tight regulation for this crucial system. Just make it such a simple thing that phenylephrine is hydrolyzed by phenylalanine hydrolase enzyme and it is converted into L-tyrosine. Here you just see the structure that OH group is transferred onto the phenyl ring and convert it into phenol. This L-tyrosine with tyrosine hydrolase enzyme is converted into L-DOPA. If you see the structure, this phenol moiety is converted into catechol nucleus. Then after the words, decarboxylase enzyme work on it and convert it into dopamine. Here you see the structure that carboxylic acid functional group is removed from the structure and afterwards dopamine beta hydrolase just transfer the OH group onto the beta carbon of this dopamine and it will convert it into norepinephrine. Afterwards, this norepinephrine by the action of phenylethanolamine and methyltransferase PNMT converted into epinephrine. Here, the amino group of this compound is methylated. Okay, so it is the complete biosynthesis of the catecholamines. These synthesized catecholamines are stored in specialized organelles in presynaptic neurons. Norepinephrine is stored in synaptic vesicles. These storage vesicles are also contained ATP, dopamine beta hydroxylase enzyme, and other proteins that helps in maintaining catecholamine stability and regulates their release. Catecholamines release occurs in response to stress, hypoglycemia, or physical activity. In neurons, action potential triggers calcium influx and causes vesicle fusion with the presynaptic membrane and exocytosis of norepinephrine. In the adrenal medulla, acetylcholine from preganglionic synaptic fibers stimulates nicotinic receptors on chromaffin cells, resulting in calcium influx and release of both epinephrine and norepinephrine into the bloodstream. Approximately 80% of catecholamines released from the adrenal medulla is epinephrine, while the remaining 20% is norepinephrine. If I'm talking about the metabolism of catecholamines, these catecholamines are metabolized by two major enzymes, that is monoamino oxidase, MAO, found in mitochondria of nerve terminals. It's deaminates catecholamines, and the second one is catecholamine catechol oat methyl transferase, COMT, found in liver, kidney, and other tissue. It methylates the catechol group. The metabolic pathway finally results in the formation of normetanephrine and metanephrine, and finally into VMA, that is vinyl mandelic acid. In synaptic cleft, norepinephrine is primarily removed by reuptake into the presynaptic neuron via the norepinephrine transporter, that is NET. Once reabsorbed, norepinephrine can be used or metabolized by MAO. In contrast, circulating catecholamines, mainly from the adrenal medulla, are broken by MAO and COMT in the liver and kidney. अगर मैं सिंपल वर्ड्स में बात करूं तो हमारी जो बायोसिंथेसिस है वो प्री साइनेप्टिक गैंग्लियंस में हो रही है। प्री साइनेप्टिक गैंग्लियंस में बेसिकली जो हमारे एमिनो एसिड है फिनाइल एलानिन, वो एल टायरोसिन में कन्वर्ट होता है बाय द एक्शन ऑफ एंजाइम फिनाइल एलानिन हाइड्रोलेस। यहाँ पे ये कंपाउंड हाइड्रोलेस ग्रुप बेसिकली इसका काम है हमारा OH ग्रुप का ट्रांसफर कराना। OH ग्रुप में ट्रांसफर होते ही ये फिनाइल मोइटी फिनॉल में कन्वर्ट हो जाएगी। फिनॉल के बाद हमारा ये एल टायरोसिन बनेगा। एल टायरोसिन फिर से इस पे एक एंजाइम वर्क करेगी, दैट इज टायरोसिन हाइड्रोलेस। वापस ये हाइड्रोक्सिल ग्रुप यानी OH ग्रुप का ट्रांसफर कराएगी और ये कंपाउंड फिनॉल से कन्वर्ट हो जाएगा कैटाकोल। कैटाकोल में कन्वर्ट होने के बाद इसका डीकार्बोक्सिलेशन होता है बाय द प्रेजेंस ऑफ एंजाइम एल एरोमेटिक एसिड अमीनो एसिड डीकार्बोक्सिलेज। जैसे ही हमारा ये कार्बोक्सिलिक एसिड फंक्शनल ग्रुप रिमूव आउट होगा, ये डोपामीन में कन्वर्ट हो जाएगा। डोपामीन फर्दर नॉरएपिनेफ्रिन में कन्वर्ट होता है बाय द एक्शन ऑफ एंजाइम डोपामीन बीटा हाइड्रोलेज। यहाँ पे जो बीटा कार्बन है हमारा, उसके ऊपर OH ग्रुप ट्रांसफर हो जा रहा है। फाइनली ये एपिनेफ्रीन में कन्वर्ट होगा। जैसे ही कैटाकोलामीन्स ये सिंथेसाइज होते हैं, वैसे ही हमारे प्री साइनेप्टिक गैंग्लियंस में साइनेप्टिक वेसाइकल्स के अंदर स्टोर हो जाते हैं। फिर हमारा जैसे कोई भी हमें एक्शन पोटेंशियल आता है इन हाइपोग्लाइसेमिक कंडीशन, इन फाइट और फ्लाइट रिस्पॉन्सेज, तो हमारे कैल्शियम आयंस कैल्शियम गेटेड आयन चैनल से इन्फ्लक्स होंगे। साइनेप्टिक वेसाइकल्स जो है वो हमारा घुलना शुरू हो जाएंगी और जितने भी हमारे न्यूरोट्रांसमीटर हैं साइनेप्टिक क्लेफ्ट में रिलीज आउट हो जाएंगे। रिलीज होने के साथ ही हमारे पोस्ट साइनेप्टिक गैंग्लियंस के ऊपर जो रिसेप्टर्स प्रेजेंट है, दैट इज अल्फा एंड बीटा रिसेप्टर्स, ये हमारे न्यूरोट्रांसमीटर्स उनके साथ बाइंड करते हैं और अपना बायोलॉजिकल रिस्पॉन्स शो करते हैं। ये बायोलॉजिकल रिस्पॉन्स अलग अलग रिसेप्टर्स के बेसिस पे हमारे अलग अलग हो सकते हैं। तो बेसिकली हमारा जो बायोसिंथेसिस हो रही है हमारी, वो हो रही है प्री साइनेप्टिक गैंग्लियन में, और जो एक्शन हमें मिल रहा है, बायोलॉजिकल फंक्शन मिल रहा है, वो रिसेप्टर साइट के बाद बाइंडिंग होने की वजह से मिल रहा है, जो रिसेप्टर प्रेजेंट है पोस्ट साइनेप्टिक गैंग्लियंस में। सो अगर हम लोग इस बात को देखें तो हमारा जो पर्टिकुलर एड्रेनर्जिक फंक्शंस हैं या बायोलॉजिकल रिस्पॉन्सेज हैं, वो अलग अलग न्यूरोट्रांसमीटर्स के अलग अलग रिसेप्टर्स के ऊपर बाइंड करने से मिलते हैं। जब हमारा पर्टिकुलर बायोलॉजिकल रिस्पॉन्स आ जाएगा तो ये कैटाकोलामीन्स वापस से रिसेप्टर साइड से अनबाइंड होंगे, साइनेप्टिक क्लेफ्ट में आ जाएंगे, और यहाँ पे इन पे दो एंजाइम्स वर्क करती हैं, दैट इज माओ एंड कॉम्ट। माओ नाम से ही हमें समझ आएगा, मोनो अमीनो ऑक्सीडेज, जो कि अमीनो ग्रुप का ऑक्सीडेशन कराएगी। नेक्स्ट कॉम्ट कैटाकोल ओ मिथाइल ट्रांसफेरेज एंजाइम है, जो कैटाकोल मोइटी के ऊपर हमारा मिथाइल ग्रुप का ट्रांसफर करती है, और ये कैटाकोल मोइटी रिकन्वर्ट हो जाएगी। जैसे ये हमारा रिकन्वर्जन होता है, ये हमारे इसके डिग्रेडेशन प्रोडक्ट फॉर्म करती है, जैसे कि हमारा मेटानेफ्रीन, नॉर मेटानेफ्रीन एंड वीएमए, दैट इज विनाइल मैंडेलिक एसिड। ये जितने भी डिग्रेडेटेड प्रोडक्ट्स हैं, ये हमारे यूरिन के थ्रू हमारे एक्सक्रीट आउट हो जाते हैं बॉडी के साथ। जो कुछ बचे हुए न्यूरोट्रांसमीटर हैं साइनेप्टिक क्लेफ्ट में, वो रीअब्सॉर्ब कर लिए जाते हैं हमारे प्री साइनेप्टिक गैंग्लियंस में, और वो फर्दर हमारे यूज कर लिए जाते हैं। ये पूरा पाथवे है हमारा फ्रॉम बायोसिंथेसिस टू द excretion of catecholamines in the body. Let's have a look on adrenergic receptors. These adrenergic receptors are also known as adrenoreceptors, are G protein couple receptor GPCRs that mediated the physiological effect of catecholamines, primarily norepinephrine and epinephrine. These receptors are key component of synaptic nervous system responsible for body's fight and flight responses. There are two main types of adrenergic receptors, that is alpha and beta, each with further subtypes. Alpha receptors are divided into two parts, that is alpha 1 and alpha 2. Alpha 1 receptors found mainly in vascular smooth muscles, pupils, and bladder. Its activation leads to vasoconstriction, pupil dilation, contraction of bladder sphincter. It works via GQ protein, which activates phospholipase C, increasing intracellular calcium level. Further, alpha 2 receptor is located in presynaptically on nerve endings or nerve terminals in the central nervous system. Its activation causes inhibition of norepinephrine release, reducing synaptic outflow. It works via GI protein, which inhibits adenyl cyclase, reducing CAMP. Beta receptors are divided into three categories, that is beta 1, beta 2, and beta 3. Beta 1 receptors found mainly in heart and kidney. Their activation results in increasing heart rate, chronotropy, increasing force of contraction, inotropy, and renin release from the kidney. It works via GS protein, increasing CAMP. Beta 2 receptors are located in bronchial smooth muscles, vascular smooth muscles, liver, and uterus. Their activation causes bronchodilation, vasodilation, glycogenolysis, and relaxation of uterine muscles. They also work via GS protein and increasing CAMP. Lastly, beta 3 receptors are found in adipose tissues and bladders. They stimulate lipolysis and relax the detrusor muscles. These are the basic receptors, their locations, and their biological functions, which are being incorporated in with the adrenergic system.